Alright, following the last explanation we gave on glycolysis, MELVISWORLD is here again to crown it up all in circumventing those incircumventable kinesis reactions. Recall we said in glycolysis, which is the breaking down of glucose to form pyruvate, which is then channeled into the TCA cycle to produce reducing equivalents, which then funneled into electron transport chain to produce energy. So as you follow us today, you will get to comprehend how those steps which were catalyzed by kinesis enzymes can be circumvented using another pathway. So, what is gluconeogenesis? It is formation of glucose molecules from non-carbohydrate sources. This as a result of low blood sugar. Now, we primarily know that this primary source of glucose to the blood or to the brain or to the body supply is carbohydrate. However, what if there is no carbohydrate source of glucose? Does it mean that we don't have any other source? This pathway is going to let you know how to get carbohydrate from other source, or rather how to produce glucose from other source other than carbohydrates. So follow us keenly as we move on. The objective of our class today is, at the end of this class, should be able to comprehend the glycolytic pathways and its clinical importance. And then the objective as well is undergraduate exam focus. Content: diagrammatic illustrations and explanations, gluconeogenic pathway and associated enzymes, regulations of gluconeogenesis. So we welcome you again, your tutor again for today, Cephas. Alright, just like earlier said, gluconeogenesis, this is the formation of glucose from non-carbohydrate sources. So when you look at this, gluco simply means glucose. Then you have new, genesis, beginning. Thus, gluconeogenesis is the formation of new glucose from non-carbohydrate source. When you begin to make or form new glucose from non-carbohydrate source, it is called gluconeogenesis, new glucose formation. Now, what are these common sources of glucose aside carbohydrate source? You have one, amino acid, specifically gluconeogenic amino acids. Then glycerol. We know that fatty acid is made up of glycerol heads and we know that fatty acid made up of glycerol head and lipids. So the glycerol heads can be channeled into gluconeogenic pathway. So all chain fatty acid, which is insignificant, and we can recall that during all chain metabolism, that is beta oxidation of all chain, we have substrates. So some of these substrates insignificantly enter gluconeogenic pathway. Now, what are the sites of gluconeogenesis? You have liver cells, kidney cells. Specifically in the kidney, recall you have proximal convoluted tubule of the kidney, specifically the nephron. Then you have the distal convoluted tubule. So, specifically in the kidney where gluconeogenesis takes place is the proximal convoluted tubule. Then, small intestine, little minor gluconeogenesis takes place in the little intestine. Okay, but before then, it's paramount you note that gluconeogenesis is absent in the muscle. Now, recall we said that glycolysis takes place in the cytoplasm of all body tissues, but gluconeogenesis is only sourced majorly in the liver, the kidney, and little quantity in small intestine. Thus, that is to say, gluconeogenesis doesn't take place in the muscle. What triggers gluconeogenesis? The primary reason why the body searches for other mechanisms to produce glucose is low blood sugar level, clinically known as hypoglycemia. When there's fall or decrease in normal blood glucose, insulin, which stimulates glycolysis, is inhibited, while glucagon is being stimulated to facilitate gluconeogenesis. Another reason is the brain. The brain's demand for glucose is low. And of course, we know that the primary source of fuel to the brain is glucose. So when there is no glucose supply, the brain seeks for another alternatives, which is the secondary source of fuel, ketone body. However, these ketone bodies are dangerous to the brain because ketone bodies can make the blood acidic, leading to ketoacidosis. And we can recall that the examples of ketone bodies, you have acetone, you have acetoacetate, you have a beta hydroxybutyrate. So we proceed. By the time we get to fatty acid or metabolism of ketone bodies, you get to know about this clearly. Alright, from the little explanations, by now you should be able to know what is gluconeogenesis, what are the primary sources of gluconeogenesis, what are the major sites of gluconeogenesis, which among the following does not accommodate gluconeogenesis. These were keenly explained in the previous slides. What is hypoglycemia, which I told you initially what hypoglycemia is here. Then, what is brain's primary source of energy? Ketoacidosis results from what? Explain. Now, it's paramount that you don't take these assessments for granted, because this neglected aspect that students avoid still comes out to confront them in exams. So, take this so serious. So, illustrations. Please, for comprehension purpose, pay keen attention to these illustrations. Now, recall that glucose at the extracellular fluid is being transported into the cell membrane by glucose transporters. And when it's relative to kidney, you have GLUT3, is example of glucose transporter, and the liver, GLUT2. So on getting inside, after being phosphorylated by glucokinase or hexokinase, it proceeds down here and move down and move down, all down and then form pyruvate. Now, our basic purpose of this tutorial is gluconeogenesis. You'll be wondering, why do I have to illustrate again glycolysis as explained in the last video? But it will interest you that the reason for that is because there were several steps which were irreversible, catalyzed by kinase reactions, which gluconeogenetic pathway is here to circumvent. Now recall here that when glucose enters the cytoplasm of the liver cell, it's being phosphorylated to form glucose 6-phosphate, and then glucose 6-phosphate isomerized by an isomerase enzyme to form fructose 6-phosphate. Then, in addition of one more phosphate group by a kinase enzyme to form fructose 1,6-bisphosphoglycerate, which is then cleaved into two by an aldolase to form dihydroxyacetone phosphate and glyceraldehyde triphosphate, which is then broken down to glyceraldehyde triphosphate down to 1,3-bisphosphoglycerates, all down by mutase enzyme adds here and then cracks it down to 2-phosphoglycerates, and then to PEP, phosphoenol pyruvate, which is then acted upon by pyruvate kinase to form pyruvate. And in the previous video we said kinase reactions are irreversible except step 6 in glycolysis pathway. So step 1, let's start with step 9 and see how this can be circumvented. Step 9, you can see that lactic acid is being converted into pyruvate. Recall that in an anaerobic condition, pyruvate is being converted to lactic acid by an enzyme known as lactate dehydrogenase, which is written here. Lactic acid converted pyruvate. But this step is reversible reaction rather. So lactate dehydrogenase can easily be converted back to pyruvate when there is demand for glucose formation. So is another source through which lactic acid can be funneled back into gluconeogenic pathway. But however, like we said earlier, the primary source of gluconeogenesis is amino acid. Now, let's take a look at this protein molecule. This is the amine group. This is the carboxyl group. Proteolysis is the breaking down of proteins into droplets. Example of gluconeogenic amino acid is alanine. Now, amino acid plus keto acid, example of keto acid, alpha ketoglutarate. A new amino acid is produced, cleaving off a modified amino acid, which is alanine. Alanine then flows into the pathway following three fates. It's either converted directly to pyruvate or converted to acetyl-CoA or converted into alpha-ketoglutarate in the electron transport chain. In the TCA cycle, recall TCA cycle takes place in the mitochondria. So probably when we are saying modified amino acid is being converted into alpha-ketoglutarate in the TCA cycle, meaning it moves directly from the cytoplasm into the mitochondrial internal membrane. So another source is, as we've seen here, that this one of the source, aside lactic acid, one of the source of generating substrate into the TCA cycle. Now you will wonder how lactic acid modified keto acids like alanine entering pyruvate, acetyl-CoA, alpha-ketoglutarate, what's their business in circumventing this pathway? What's their business in generating glucose? Recall that once this substrate, lactic acid modified keto acid, flows into the TCA cycle, you notice one thing is a progressive steps, and when it's being gotten to malate, malates is readily converted to oxaloacetate and isomerized back to marate. Now, oxaloacetate cannot permeate the mitochondrial membrane, so therefore, oxaloacetate, the malate itself, can be channeled outside the mitochondrial membrane as malate and then converted or isomerized to oxaloacetate. That is, if the gluconeogenic substrate are coming from lactic acid modified ketone bodies, these procedures will be necessary. However, recall that pyruvate has three major fates. Under aerobic condition, converted to acetyl-CoA to carbon molecule, anaerobic condition, converted to lactic acid 3-carbon molecule, and then under normal circumstances, pyruvate is also converted to oxaloacetate by pyruvate carboxylase, addition of carbon molecule, making it 4-carbon molecule. So once that is done and the body is short of glucose, every other procedures, every other pathways are shut down. The already formed pyruvate will then be channeled into oxaloacetate, which will then be isomerized to malate and be funneled outside the cell membrane or the mitochondria to produce oxaloacetate. Also, from modified ketone bodies, from modified keto acids and lactic acids, we imagine them entering pyruvate directly. So, if there is low blood glucose, the modified ketone body wouldn't bother entering the TCA cycle by going through the cell membrane or the mitochondria. It will first of all flow into the pyruvate, and then the pyruvate will be converted to oxaloacetate by pyruvate carboxylase, which will then be isomerized to malate, pushed outside the mitochondrial membrane, and then formed oxaloacetate. However, oxaloacetate is a 4-carbon molecule but need to be cleaved off 1-carbon molecule so as to form pyruvate. So PEPK, phosphoenol pyruvate kinase, cleave off one carbon molecule to channel oxaloacetate to the formation of phosphoenol pyruvate kinase. This arrows is pointing upward. So once phosphoenol pyruvate kinase is formed, then what happens? It comes over here. You can see the pathway is moving upwards now rather than going back. So looking back here, there are enzymes that helps to circumvent this irreversible pathway. Reversible step 9, pyruvate to pyruvate, conversion of phosphoenol pyruvate to pyruvate by pyruvate kinase. So example of the enzymes, pyruvate carboxylase. When you look at this, oxaloacetate is 2 carbon molecule, and if however it must be converted to pyruvate, it must be added another carbon molecule. So pyruvate carboxylase can actually come in and add one more carbon molecule to acetyl A, and then push it back to pyruvate, and then pyruvate be converted to oxaloacetate by carboxylase enzyme, then move out and then enter the pathway. Also, the irreversible steps again of pyruvate, which is catalyzed by pyruvate carboxylase, Pyruvate decarboxylase can come and cleave off this one carbon molecule and it will form back pyruvate. However, the major glycolytic enzyme circumventing pyruvate irreversible reaction is pyruvate phosphoenol pyruvate carboxy kinase, which we see here. So the pathway begins to move up, getting to steps 3. Recall we said step 6 is a kinase reaction but can be circumvented. So getting to step 3, you have normally supposed to be 1,3-bisphosphatase converting glyceraldehyde, converting this reaction. But here now, this enzyme comes in to play its role by cleaving off one phosphate molecule, and the name is 1,3-bisphosphatase. And then it moves on progressively, getting to this step. This step is reversible. So 1,3-bisphosphatase cleaves off phosphate group, and then once it cleaves off phosphate group and then forms fructose phosphate, which then is being isomerized to glucose phosphate. However, glucose phosphate reaction, which was achieved when hexokinase or glucokinase phosphorylates it, is irreversible. So the next thing the body does is to channel glucose phosphate into the smooth endoplasmic reticulum of the liver, because there is an enzyme domiciled there known as glucose phosphatase. So glucose molecule enters the pathway, and as it's entering, glucose phosphatase cleaves off the phosphate group, and then glucose is coming out as free glucose and then transported outside the cell. In this case, glucose is made available for the cell. Also, like we said, insignificantly, here is fatty acid, here is glycerol head, here is lipid tails. So once there is lipolysis, the glycerol head is being separated from the fatty acids, and glycerol head can be added phosphates by glycerol kinase and then form dihydroxy acetone phosphate, which then can enter this pathway. It can as well be merged together to form fructose 1,6-bisphosphoglycerate. The free fatty acids can then flow down. Free fatty acid coming down and then enter succinyl-CoA. Succinyl-CoA then be channeled up to malate. Malate be pushed out the mitochondrial membrane of the mitochondrial membrane and then isomerized back to oxaloacetate, and then phosphoenolpyruvate carboxy kinase converts it back to PEP. So these are basic steps of gluconeogenesis. So hormones involved: glucagon, epinephrine, norepinephrine, cortisol, thyroid hormones, growth hormones. Key enzymes you need to know. Key enzymes of gluconeogenic pathway: pyruvate carboxylase, phosphoenolpyruvate carboxy kinase, fructose 1,6-phosphatase, and their key step, the stages where they are at. Pyruvate carboxylase acts as step 9. Then phosphoenolpyruvate carboxy kinase acts as conversion of oxaloacetate to phosphoenolpyruvate, also step 9 stage. Then fructose 1,6-bisphosphatase, which catalyzes the conversion of fructose 1,6-bisphosphate by cleaving off one phosphate group. Okay, now we are going to look at regulations of gluconeogenesis. It's being regulated. 1. Allosteric regulation, hormonal regulation. Key enzymes involved, as said earlier: phosphoenolpyruvate carboxy kinase, pyruvate carboxylase, fructose 1,6-bisphosphatase, heavily regulated by fructose 6-phosphate. By fructose 2,6-bisphosphate. There is a little error in typographical error. Fructose 2,6-bisphosphatase. Functions of this enzyme: phosphoenolpyruvate kinase. Phosphoenolpyruvate carboxy kinase catalyzes conversion of oxaloacetate to phosphoenol so as to promote gluconeogenesis. Please pay key attention at this point. Your examiner might ask you this question. So the function of PEP is conversion of oxaloacetate to phosphoenolpyruvate. PC, pyruvate carboxylase, catalyzes the irreversible formation of oxaloacetate from pyruvate, which promotes phosphoenolpyruvate formation during gluconeogenesis. Thus, this inhibition prevents substrate production, which is oxaloacetate, which would have favored gluconeogenesis by action of PEP. So, like we said, when we are talking about the inhibition of these enzymes, Inhibiting any of these enzymes can prevent gluconeogenesis from occurring. So let's proceed. Fructose 1,6-bisphosphatase catalyzes the formation of irreversible reaction of phosphofructokinase reaction in glycolytic pathway, which led to formation of glucose 1,6-bisphosphate substrates. Now, regulation. Allosterically, citrate activate fructose 1,6-bisphosphatase. Citrate activates fructose 2,6-bisphosphatase, and AMP are inhibitors of fructose 1,6-bisphosphatase. Pyruvate carboxylase is stimulated by acetyl-CoA and inhibited by ADP. That is to say, high level of acetyl-CoA, pyruvate carboxylase will be stimulated to convert much acetyl-CoA back to pyruvate by adding carbon molecule. And it's being inhibited when there is high ADP. High ADP simply tells you there is low energy, there is low ATP. So the body, the enzyme pyruvate carboxylase is being inhibited so as to produce more pyruvate, which will then be converted to acetyl-CoA, which finally enters the ECA cycle to produce reducing equivalent that then be funneled to electron transport chain to produce energy. So hormonal regulations, here you have cortisol released when blood glucose is low, and during chronic stress activates specific genes that stimulate the synthesis of the gluconeogenic enzymes PEP, PC, F1,6-bisphosphatase. Glucagon dephosphorylation reactions stimulates PEP and PC. Assessment. You get to the site and get these materials. Look down on them. It's not enough to just watch these tutorials. Assess yourself whether or not you can be able to comprehend what I have explained to you, and get yourself ready for the exam. Thanks. Wishing you all the best of life.